It's okay, no?
Yes, I think you can start.
Okay. Hello, everybody. I am Max Bonomi. I'm here with two other developers of Plumed Software, Giovanni Buzzi and Gareth Ribello, as you can see in the camera. I want just to warn you that this lecture will be recorded. So if you don't want to share your camera or you want to turn it off, you can do it. It's very nice if we see some faces, because I don't know you, but we are kind of confined these days in our apartment, so it's good to see a few new faces. Otherwise the lecture will be recorded and put on YouTube. I will send the link after this. We will send the link after this lecture. So this is the first of a series of what we call PLUMED Masterclass. It's a series of classes to teach you some basics about how to use this software, but also some more theoretical and theory basis to do analysis of MD simulation or method to enhance, accelerate sampling in molecular dynamics simulations. You can stop me at any time if you have questions. You just have to switch on your microphone and you can interrupt it. There's no problem at all. Today, for this first class, we will talk really about the basis of the software. I will show you a few slides to introduce the software, its basic structure, what it can do, and then I will tell you what is the content of this lecture. Just to remind you how these lectures are organized for every subject, so we call it a class. There are actually two lectures. One is the introduction to the problem that we are going to address with some theory, depending on cases. And the instructor today is me, in this case. I will show you the exercises that you will have to do together or alone during this week. So you have 1 week to solve the exercises, and then we will see each other again on Zoom next week, where we will solve the exercise together. I will show one solution. Maybe some of you can will show what they did during this week. During this week, you will interact with us, the instructor, and with the other students on the Slack channel that you should have access to. So today, as I told you, is about the basics of the Plume software, how to use it, its syntax, and how to use to analyze some molecular dynamic simulation that you have already performed. And the software, the Plume software, is an open source and freely available software. It's a library written in C++ that can do several things. The basic idea is that you have a collection of methods to accelerate sampling of molecular dynamic simulation, what we call enhanced sampling techniques, and method to calculate or free energy as a function of some descriptor of the system, what we call collective variables. And we will see some example in this lecture. And the same tools and the same collective variables can be used to analyze pre-existent molecular dynamic simulation, which is actually what we will do today. And the main developers of the software are four: Gareth Ribello, Max Bonomi, me, Carlo Camilloni, who is not here today, and probably it will be in the next few lectures, and Giovanni Buzzi. And we are all, as you can see, distributed in almost equally spaced in Europe. And the functionality of Plume, the basic functionality is to calculate these collective variables. So this descriptor, coarse-grained descriptor of the system. It can be something very easy and very simple, like, for example, the distance between two atoms, or something a bit more complicated. Sorry. Like, for example, if you have a protein, you can quantify the secondary structure content of some part of a protein, how much of an alpha helix you have in one region of a protein, or a beta sheet. Or these are variables tailored a bit on biological problems and biological systems. You have collective variables also for more material science problems, like crystallization, for example. And I think Gareth maybe will show something in future classes. And you have new collective variable based on dimensionality reduction or machine learning technique, and you will probably see in more advanced lecture. And so the basic functionality is to calculate on the fly during a molecular dynamic simulation these variables, and to use them to accelerate the sampling in combination with methods like metadynamics, umbrella sampling, or steer molecular dynamics, which are not the subject of today's lecture. And but these add a bias potential to your standard molecular dynamics simulation, which is typically a function of the collective variables that I've just talked about. The third functionality of Plumed is to do analysis of these CV, calculate these CVs on existing trajectory, and we have all the tools to post-process all the most popular formats for molecular dynamics simulation, like XDC or DCD and other format. So in this sense, Plumed can be used as a standalone utility or in combination to many popular molecular dynamics code. This part is to do the analysis on the fly or to implement these enhanced sampling approaches. You need to work together with a molecular dynamics code. And as of today, we work with several codes like GROMACS, NumD, LAMPS, OpenMM, ASMD, and the list is growing with time. The code has been restructured a few years ago in order to be extremely modular. So we have really a core of functionalities and then a series of modules and which implement some specific functionalities. For example, we have different collective variables that I've talked about, these different enhanced sampling techniques, or different, let's say, modules to perform different and specific functions. And these are not, in many cases, are not written by ourselves, are for developers, but there are many other contributors in the community that have written parts of the Plumed code to do something specific that they needed for their research. So if you one day you want to look at the code, you will see that around this core of Plumed, there are a series of different modules which contains code to perform, for example, integration of experimental data in a molecular dynamics simulation, and this module for integrative structure and dynamical biology, or specific collective variables like this permutation invariant CV, or variables that describe a pathway of reaction between two different states, and so on and so forth. And this has been, as I told you, this module has been contributed by many different people. If you are interested in learning more about the code, there are several places where you can look at. Our main reference is our website, plumed.org, but you can have a look at the GitHub if you want to look at the source code, or follow us on Twitter and the different news. If you encounter problems in the future, we have a user and developer mailing list, and many tutorials already are out there. So as far as today is concerned, what we will do now is have a look at the exercises for this first class, and we will look at the exercises together. And to do this, we will go to the Plumed website, plumed.org. And if you look at the main page, you need to click on the Masterclass tab, and you will see in the pages, in the page that you open, there will be a table with all the classes, present and future classes. You have to click on the topic of this lecture, so Plumed syntax and analysis in red, and you will end up in a new page. I will do the same in 1 second, and I will show you the exercises. And as I told you, you have 1 week to complete them. All the question and discussion can be done on the Slack channel for this masterclass. And after this lecture and the next one is completed, we will post on YouTube the link to this recording, and you have just to click in this table the date of the lecture, and you will end up on YouTube. So now I will do exactly this. I will go to our website, click Masterclass, and I select the first one. You should all be there, I imagine. Please stop me if you have problems at this point, just please ask me. So this is how it's structured this today's lecture. We are going to learn how to write a simple Plumed input file to do some post-processing and analysis of trajectories that I I ran for you some time ago. We will learn basic commands, for example, to calculate distances between atoms of simple properties of the system along the molecular dynamics simulation. And then the exercise will become a bit more complex, and we will see, for example, how to fix some discontinuities in the trajectories due to the periodic boundary condition that are sometimes make your protein or your system broken and not entire. And we will learn more sophisticated analysis as we go forward in the exercise. The first thing that you will have to do this week is to install the software. We made this pretty easy for you because we install everything with Conda. If you are not familiar with Conda, is a way, basically, very easy way to create separate environment. And in this environment, you can install any software that you want, and you can have different environments with different versions of each code. So it's a very convenient way to keep things very organized. So you need to check if your Conda is installed. Otherwise, we provide instruction how to install this on your machine. Then we create an environment for this PLUMED Masterclass. Most of this environment, if you will attend the future lectures, it will be useful also for the future lectures. So we call it PLUMED Masterclass with the environment, and we activate it. And once you've done this, you can install all the software that you need with this line. So once you've done this, you are really good to go and to start the exercises. So as I told you, Plumed works in two ways. One is on the fly during the molecular dynamics simulation, and the second way of working is as a standalone set of command line tools. And you can look at the Plumed executable, and if you type plume --help, you will have a list of all the possible commands, standalone commands that Plumed implements. And the main command that we will use in all these exercises is called Plumed Driver. And this enables analyzing an existing MD trajectories in many different formats. So this is just to give a brief introduction of the main tool, Plumed Driver, that we will use this week. So you don't have to run the molecular dynamics simulation. I providing the data on GitHub. So you need just to download all the data here from GitHub. And you will find in this repository different things. The most important information is in directory called data. You will have two files. One is a reference conformation of your system. This is a membrane receptor with this strange name. Here is a serotonin receptor, and this conformation is with one ligand somewhere in the, I think, in the extracellular part. You will see the ligand. There's no water, there's no membrane here. I just eliminate this from the PDB, from the trajectory. You have just your protein and the ligand, and then you have the same trajectory. This is in the folder called data. Sorry. And then you will have another folder which contains an instrument, Jupyter Notebook, that we will use for the analysis of the, let's say, of the output of our Plumed software. And I will show you where we use this. As I anticipate, many software for molecular dynamics produce a trajectory that is discontinuous, in the sense that your system is sometimes broken in different pieces. And this is because the simulation is always in a periodic box, and the software, most of the time, Chromax in this case, doesn't care about in which copy of your periodic cell the system is. So maybe you have some atoms in one copy of a cell, other atoms in another copy. In this sense, the system, the trajectory is discontinuous, because you might see some atom jumping from time to time from one cell to another. And we need to keep this in mind in order to do our analysis correctly. So you have a trajectory, you have a PDB file of the system that you can have a look. And the other thing that we need in order to do analysis with Plumed is to create what we call a Plumed input file, which is a list of instruction of what to do with Plumed to analyze the trajectory. So if you have no experience with Plumed, I just pasted here an example of this Plumed input file here. So it's very simple. And for example, here what we do is we define a distance between two atoms, atoms 1 and 10. This is this numbering is it's always start from 1, and it identifies the atoms, for example, as in the PDB file of your system. So it's the way we identify atoms with their index here. So we define a distance between two atoms, and we call it D, so we can use it later. And then we define other collective variables like a torsion, and we define with 4 atoms. And what we do, very simply, is we want to print the value of this distance to this file, which we call Colver, every 10 steps of your MD trajectory. And then we want to print of another file these 2 variables every 100 steps. So the things is very easy. We define this collective variable, we give them a label or a name, and in this case, we just print their value along the trajectory. So this is a structure of the input file, this very simple input file. If you don't know what these things are, you can at any time click on this keyword, and you will end up in our manual when you have a basic description of what is the distance. In this case, it's pretty intuitive, so I don't think you need to have a look. But in other cases, if you don't know what is printed here, you just click and you will have a reference to the manual. In some cases, you can even go on top of this keyword and have a description of what is this argument, for example. This is typically for the option. So for the main keyword for the action, you can go to the manual, and then you have a description of the different argument here. Okay, this is just to give you an idea of what is a Plumed input file. In the exercise that I will show you, you will have to write by yourself a Plumed input file to do something that I will explain you. Before continuing, I want just to make a small comment. The Plumed has its own units for all the different quantities. For example, the length or distance, for example, is always measured in terms of nanometers, energy in kJ per mole, time in picoseconds, and this is probably what we need for the exercises today. So keep in mind that these units are fixed no matter what molecular dynamics code you're using. Okay, so we start with the first exercise that you will do is basic computing some basic collective variable and printing them on file. And to do this, you need to create your own input file. You have, let's say, a template here. Every time, so we give in the beginning, we are giving you some suggestions. Just please don't use this as it is, because you see there is this yellow keyword, fill, which means here you have to do something. You cannot use this as it is. You just need to put some information there. And the objective of this exercise is to calculate 2 collective variables. One is the gyration radius of the atoms, C alpha atoms of this protein, only in the first 40 N-terminal residues. This is if you look at the structure, you can look here at the structure of the system. These first 40 residues is an extracellular flexible loop of this receptor, and you want to study the dynamics of this part. So, for example, you want to see if it's very compact, or elongated, or it samples different conformation. So we want to calculate, as a measure of the overall shape of this part, we want to calculate the gyration radius of this fragment. And the second variable that we want to look at is the distance between the C alpha atoms of just the first and last residues. kind of an end-to-end distance of this flexible part of the system. So to do this, you have to fill in and complete this input file following the instruction that I put it here. Once you have done this, you need to calculate this quantity on your trajectory that you downloaded from GitHub. And the command is called Plumed Driver, and the syntax is written here. This plume.dat is the name. You can put whatever name you want, but it's the name of this input file. So you have to put this in the plume.dat input file and complete. Once you've done this, you can run the driver, and you will have as output one file, which is called Colvar, because we told Plume to call it Colvar. And this Colvar you will have for each frame of your MD trajectory the value of the radius of gyration and of the distance. At this point, we want to do some analysis on this output file, and we have prepared a basic Jupyter notebook that you can find again in our GitHub. But just to remind you, this one. So this is the basic data that you need for this class is contained here. So also the GitHub, the notebook is in this GitHub. So let's go down. This notebook is, you will use it and adapt it in all the exercises, but this one is already basically ready for these exercise, and it's reading the Colvar file as it comes out from Plumed, and then you visualize some quantities, for example, the time series of a gyration radius, the time series of a distance, and so on and so forth. And then here, at the end of the exercise, there are some questions that you need to answer after analyzing the output of Plumed. Do you have any question so far? No questions? Okay. So this is the first exercise. Is very simple, two collective variables and an analysis of a trajectory that we provided for you. In doing this, you will see that there is one kind of boring operation, which is defining the list of C alpha atoms of these 40 residues, which means filling in with this line, with 40 numbers, which is the atom index of these atoms. And this might be boring, and sometimes it's not 40 atoms, it's 1,000 atoms. So we have some advanced selection tools, and in the exercise 2, we will learn how to use these selection tools. And the keyword that the special keyword in Plumed to activate this functionality is called molinfo, and it requires a PDB file to learn something about the structure and the topology of your system. For example, this is a protein, so there are residues. Each residue has a certain name, and if you don't read the PDB file in Plume, you don't know about these properties of the system. So what we are going to do in these exercises is divided in two parts. In the first part, we want to calculate the value of a backbone dihedral angle. So if you're not familiar with proteins, here you will see what I mean. This is the backbone of a protein built of some special atoms. And what we want to calculate is the dihedral phi of this residue, which is called I. So the dihedral phi is defined by a set of 4 atoms, which are highlighted here in red. So this is one way in Plumed to define a dihedral is specifying a list of 4 atoms. So this is one thing that you can always do by looking at the PDB file and searching for the index of the 4 atoms which are needed to define dihedral. And this can be boring, especially if you don't know very well which are the atoms involved. The second possibility is to use this functionality provided by molinfo that allow you to basically, without specifying an index, 4 indexes, to specify directly the dihedral phi of this residue. So this exercise is very simple. We focus on residue number 2, and we specify the dihedral in 2 ways, as a list of 4 atoms or using this functionality. So you have to click on more info and see what is the way to specify dihedrals in Plumed, and specifically this dihedral file. And then you complete this template with the 2 torsion, so the 2 dihedral, specified in the 2 different ways, and you reanalyze the trajectory again using our Plumed driver. If you've done this exercise correctly, you can look, or you can use the notebook to visualize the Colvar file. You will see that the 2 definitions are identical, and the time series of your variable, collective variables, is identical. So this is the first part, sorry, of the exercise. In the second part, we do something a bit more exotic, and we use 2 other libraries, Python libraries, maybe you are familiar with. They are called MD Analysis and MD Trudge. So we borrow from these 2 libraries the way some advanced selection tools capabilities. So we use the way the libraries select atoms to specify atoms in Plumed. And to showcase this, to test this capability, we want to redo the first exercise, the one in which we have the gyration radius and the end-to-end distance. But this time we don't put a list of 40 atoms explicitly. We use these MD analysis and MD trudge functionalities to automatically select residue from 1 to 40 and name and only the C alpha atoms of the system. So if you're not familiar with MD analysis and MD trudge and their syntax, you have to consult the manual. It's very simple, actually. It's mostly completed here. But in this way, you have a quick, you can select these atoms without putting explicitly the list of atoms. And to check if you've done this correctly, you have to compare with the result of exercise 1, in which you put these indexes explicitly. Okay. The third exercise is about defining virtual atoms. Virtual atoms are atoms, let's say, that are defined in terms of the position of other atoms. The easiest example is the center of mass of the system, or the geometric center of a group of atoms. So this can be regarded as a virtual atom, and to define the position of this atom, you have to define a list of other atoms and the position of the other atoms. So in this exercise, we will learn how to define and use the geometric center of a group of atoms. And to do what? We want to calculate 2 things. So this protein has some special things attached to residue 24 and 32. There are some sugars that are attached. These sugars are actually called glycans. They are very important in biology. And they are polymers that are attached to sometimes to certain protein amino acids. So if you look at this PDB file, you will see that attached to residue 24 and 32, there are some long polymers. These are the glycans. What we want to do is to calculate the distance between the center of mass of the 2 glycans and the serotonin ligand. So we want to check if this ligand, serotonin, is staying closer to one glycan or the other. So to do this, we have to define first the geometric center of the serotonin in terms of the list of indexes of the atom of serotonin, and then we define the same thing for the first glycan and the second glycan. At this point, you can reuse, you see, I label these at G1 and D2. You can use this label as they were atoms, so you can use, you can define a distance using what you defined before in terms of the position of other atoms. And then you print all these to a file, these 2 variables to a file. Once you've done this again, you can post-process the trajectory and calculate these 2 distances, and you can answer to the question if serotonin is staying closer to the first or second glycan on average, let's say, overall during the molecular dynamics simulation. We are halfway, so now things becomes a bit more complicated. Exercise 4 is about how to fix the fact that the trajectory as it comes out from the molecular dynamics code is broken by periodic boundary conditions. And there are certain collective variables that are able to, let's say, to fix these discontinuities automatically. For example, the distance that you've seen here and here, or the geometric center, in most cases fixes the BBCs, if not always correctly. So you don't have to do anything in Plumed, because the information about the cell is contained in the XTC file. And so Plumed is able to reconstruct a continuous piece of a protein inside the Plumed code. In other cases, it's a bit more complicated. So you have to specify manually what are the, let's say, the list of atoms that should be made continuous inside Plumed. And there is an action, a keyword to do this, which is called whole molecules. You can click here and go to the manual to have a look. The exercise is the following. We want to estimate what is the secondary structure content of these 40 N-terminal residues of the receptor, which I told you it's a loop, it's pretty flexible, but this is a very long simulation. So from time to time, this loop populates alpha helix or beta sheet. So we want to estimate how much of alpha helix or how much of beta sheet conformation are populated during the MD simulation. And we have 3 collective variables to do this implemented in Plumed. One is alpha RMSD, and the second is para-beta RMSD, which measure the parallel beta sheet content, and anti-beta RMSD to measure the anti-parallel beta sheet content. All these collective variables require that the part of the protein that you are examining is already continuous, in the sense that PBC's discontinuities have been fixed. So what you need to do is to do this manually. So you use this all molecules keyword to make the first N-terminal residues entire, let's say, or whole, and then you can calculate the alpha RMSD and the para-beta RMSD and the anti-beta RMSD. So now there is one other keyword which is pretty useful, because we don't care about the beta sheet if it's parallel or anti-parallel. So what we can do is sum the anti-parallel and parallel content. So we want to sum these 2 collective variables in a new one, which we call the overall of the total beta sheet content. So what you should print at the end of the day is just the alpha helix content of the first 40 residues and the total beta sheet content of these residues, after you combine the 2 variables here. So if you do this, and again, if you reanalyze your trajectory, then you can quantify what is the population of alpha helix or beta sheet during your simulation, and answer to the question if one is more populated than the other. Do you have other questions? Nope. Okay, we have 3 more exercises. The exercise 5 is about using the RMSD as collective variable, so the root mean square displacement with respect to a reference conformation to measure some conformational changes. And if you know what RMSD is, you should not forget that there are 2 operations to calculate an RMSD. The first one is that you have to align one conformation to a reference, and then to calculate how different are the 2 conformations. So if it's the same conformation, but it's just translated and rotated, once you optimally align, the displacement between the 2 conformations is 0. So the first thing that you need to do is do this optimal alignment between two conformations. And then you can calculate the displacement. In Plumed, you can specify a set of atoms to do this optimal alignment and a different set of atoms to calculate the actual displacement. And in this exercise, we will try exactly this. So we want to calculate the RMSD of these flexible 40 N-terminal residues after you align the system on the rest of the protein, so on all the other residues. Why we want to do this? Because I can anticipate that the other residues are pretty stable during the simulation because they are in the transmembrane region. So we want to align the whole system on the stable residues, and then we calculate the RMSD only of the flexible part, the displacement only of the flexible part. This is one CV. The second CV is actually, we calculate the RMSD of the stable part to check that it is actually stable. So in order to do this, you have to work a bit more on your own and looking at the manual here at the RMSD page. Let's say the key here is that you have to create two different PDBs of the system. And in each PDB, you have to mark the atoms that you want to use for the alignment and the atoms that you want to use to calculate the displacement. How you do this, you need to have a look at the manual and see how, and to learn how to mark atoms for alignment and displacement in the PDB. Also, you have to check in the manual if these periodic boundary conditions are automatically taken care of when you use RMSD, or you need this option that we learned in the previous exercise. If you do this and you analyze the trajectory correctly, and if you analyze the trajectory, you can answer which is the more flexible part of the system and which is the one that deviates more from the starting conformation during the simulation. Okay, we are almost at the end. The exercise 6 is related to exercise 5 in some way, and we want to learn how to align during the course of the simulation or after in post-processing, how to align a conformation to a template. In this case, let's start from our specific case. We have a membrane proteins embedded during the simulation in a lipid bilayer. And typically, the initial conformation is oriented in such a way that the lipid bilayer is parallel to the XY plane. This is most always the case in case when you do MD simulation of membrane protein. And you can check if you look at the PDB. What we want to measure during the simulation is the position of the ligand, the distance of the ligand from the lipid bilayer. So the vertical, basically it's the vertical position of the ligand with respect to the lipid bilayer. And if you think that the lipid bilayer is parallel to the XY plane, you can use the coordinate, absolute coordinate of, I don't know, the center of mass of the ligand to measure how far is the ligand from the membrane bilayer. This is okay in the beginning because the system is oriented in a certain way. But during the simulation, you have two things. The system can translate from its original position, and also it can be broken by this periodic boundary condition. So you cannot really use an absolute coordinate during molecular dynamics simulation or after without doing some post-processing. So the post-processing that is required is explained here. If you want to use absolute position, the first thing that you need to do is to make sure that the protein is not broken by this periodic boundary condition. And you have learned how to do it in the previous exercise. The second thing, you have a serotonin. You have to check that you have to enforce, let's say, or that serotonin is not broken by periodic boundary condition. And also it is, let's say, defined in the same simulation cell, not in a copy, of the protein. So everything, let's say, is wrapped, is continuous and wrapped inside the starting, let's say, cell of the system. And we have an option to do this wrapping, which is called a wraparound. The third, now that everything is continuous and let's say in one cell, we have to align this protein, just the protein, to our initial template, which is the PDB file. If we do this at any time, any frame of a simulation is made a whole in the same cell aligned to the initial conformation. And in this situation, you can actually use safely the position of the center of mass of a small molecule to define how far is the serotonin from the lipid bilayer. And you can do this using the position collective variable. Now, this is a kind of a complicated set of instructions that you have to do on your own. And it's basically these three, you have to play with these three action in Plumed. But there is a way to check that you've done all this transformation correctly. So imagine that you have completed the input file with these three set of instructions. Then you can print out to a different file, I call it align.grow. You can print out the conformation of the system after all the operation that Plume is doing. So you want to check that you've done things correctly. So you want to post-process your original trajectory, do all the transformation in Plume, and print out the conformation after the transformation. And this is what you can try with this suggestion here. Another things that you can use to check that you've done things correctly is once you have done all this operation that I just described, and you define as collective variable the Z component of the position of the center of mass of the ligand, and you calculate the time series of your CV, this should be continuous, first of all, and defined in a certain range, which I wrote here, which is basically the extracellular domain of this system. If you see that it's not continuous or in another range, it means that something is not, it might be wrong. Okay. Last exercise. The last exercise, since we have a small molecule and a protein, we want to check what is the propensity of this small molecule, this ligand, to bind to this flexible part of the receptor. And we want to check if there are some hotspots where binding is favorite. So to do this, there are no template input file here. You have to do everything on your own. But I will tell you what you need to do. So you need to compute the fraction of bound conformation over the total number of frames in the trajectory that you downloaded from the GitHub. This is the first thing. And then the second task is for each individual residue and glycan, you have to, let's say, quantify the fraction of bound conformation per residue and per glycan. So the first thing is the overall fraction of bound conformation. The second is defined at the level of each residue and glycan. And while doing this, you need also to print out all the bound conformation to a new file, all the bound conformation and the bound conformation per residue and glycan. So you can have a look and check that things are done correctly. And to do this exercise on your own, there are some advices here. So the first is we define in an arbitrary way a bound conformation, a conformation in which there is at least one pair of atoms of the ligand and of a protein that are closer than 0.4 nanometer. So this is arbitrary. The second is you can use any collective variable that is defined in the Plumed manual. So you check, you go here and you have a look, collective variables. You have a long list of variables that you can use. It's up to you to choose wisely. If you don't like this variable, or if you think you can do it better with other variables, there is a way to define your own collective variable directly in the Plumed input file by using the custom action. So you have a look at this description here, and you will explain how to define your own collective variable. In the end, you can do this exercise as you like, but keep in mind what the goal should be reached and to compute this bound fractional bound conformation overall and per resident glycan. So these are the 7 exercises. I think they are of increasing difficulty that you need to solve by during this week. If you have problems, just we can discuss and on Slack channels with us, instructors, or among you. And I think we have a few minutes. If you have really questions, I'm happy to answer to them.
Hi, I have a question.
Yes.
How one would choose a CV in general? Like, how do we decide what CV we have to choose, whether it is a distance or RMSD or any angle?
This is up to you to the question that you have to answer is how many conformations are bound, how many conformation of a let's say of a ligand are bound to these 40 N-terminal residues. And the definition of being bound is there is at least one pair of atoms that is closer than 4 angstrom. So given this definition, you can choose optimally any collective variables that enables you to answer to this question. There are more than one. The trivial one is the distance, but which distance between which atoms? But you have to remember that the definition of bound is given in terms of distance with a cutoff of 4 angstrom. This is the only information that you need to answer to the questions. How to arrive is up to you. This exercise is exactly to stimulate you to think of a problem and translate it in choosing certain collective variables. So I cannot answer for you.
Okay.
But we can discuss more during the week on Slack. Other questions?
All right. Thank you.
No problem. No more. Yes.
I have a quick question about the software in general.
Yep.
Is it possible to use it with Desmond?
I think not at this time. I think they have their own implementation of some enhanced implementation that we have implemented, but their own native. But I'm not sure there is an official patch for Desmond.
Okay.
I can investigate. I will answer to you in the channel this week.
All right. Thank you.
No problem. No other question?
Hi, there's also one more quick question. Like, do you
Yep.
have to define multiple distance calculations if you have the multiple molecules of the same type? I guess this is only applicable for the small molecules instead of like proteins.
You can. In this specific case, you have one copy of the small molecule in the simulation box. So you don't have more than one serotonin at each time.
Yeah, but if you have more molecules in your own system, but if it's the same type, like, can you define distance based on the atom types instead of like just the atom numbers?
Yes, you can, but you have to use some advanced selection tools, which translate these atom types in atom numbers.
Oh, okay. Thank you.
You're welcome. Please have a question, even if stupid. There are no stupid questions. Is it clear how you should proceed with the exercise this week? Okay, I suggest you can start because you can do them whenever you want, but you can start as soon as possible, so you can interact more with us or with you in between you, as you prefer to organize the work. Especially for the last couple of exercises, which are a bit more difficult than the first 2 or 3. Okay. If there are no more questions. I think we can stop here for today. We are in touch this week, and we will do the correction together in 1 week, same time. Garrett, you want to add something, or Giovanni? No.
Just some threats, you know. You will get out of this as much as you put into it. So, you know, if you don't do the exercises, it doesn't affect us a great deal. But it does affect you because you maybe won't understand. So please try the exercises. Please ask questions if you have questions. There is no such thing as a stupid question, but that is what we're here to, that's what you're here to do. You're here to do the exercises, not just to listen to Max or me talk about Plumed.
Perfect.
Maybe I have one question. So this week we're only using the standalone Plumed codes. Are we going to need a compiled version with Chromax or something like along those lines?
Not for this week.
But
you won't need Chromax. In the future lectures, yes, but we will provide. Probably Giovanni can answer better than me. We can provide them via Conda.
Yeah, I think for class 2, it will also be without doing MD, if I remember correctly from the Gareth plan. But class 3, we will use Chromax.
all right.
I will provide executable on Conda as well.
Okay. Would a compiled version with version 2.5 work, for example?
I would suggest to use exactly the version that we provide on Conda, because yeah, probably most of the exercise can be done also with older versions, but it's easier if everyone is using the same version.
Okay, thanks.
Okay, so I think it's time to go back to work and to start the exercises. Okay. We'll be in touch this week on Slack, and we see each other next week. Okay? Bye. Bye-bye.
Ciao a tutti, bye bye.
Bye.